All right, everybody, welcome to Inner Plant Structure. This is Hort 300. We're going to be diving into inner plant structure. We're going to start with leaves and stems. Okay, so far we've gone through sort of the outer plant parts. Now we're going to dive into the inside and sort of the anatomy of these different parts of the plant that are in the inside. We're going to look at what they are, what their functions are, how they all work together. And the reason for this is so that we can have an understanding of how plants function and what the inner functions actually are. And with that knowledge, it helps us to understand what could be going wrong with the plant, and also helps us understand, so diagnosing pests, diseases, and abiotic plant disorders. Abiotic plant disorders being things that are not caused by pests and plants, things that are caused by excessive sunlight, excessive heat, excessive cold temperatures, excessive water, etc. Right? And there's a lot of plant disorders that occur that look like they're disease and pest related, but they're actually abiotic plant disorders. So understanding how a plant works, again, helps us to be able to diagnose issues with the plants, and also helps us to do a lot of really cool things with plants. Okay, so we'll dive into it, right into the leaf itself. So looking at the leaf structure, of course, we have this wonderful diagram of a leaf. This is a little section cut out so that we can see what that leaf structure looks like. And the main function of the leaf itself, is photosynthesis. Okay, main function of the leaf itself is photosynthesis. So we're going to dive into a quick video on photosynthesis itself, and then we're going to dive into each one of these parts of the inner plant, these different types of cells, and sort of what they do, what they are, and what their function is. Okay.
The amazing process of photosynthesis. A look around at nature is an absolute treat to our senses. We all enjoy the colors and fragrance of plants, but seem to forget about their important contribution. Plants are an absolute necessity for life on Earth. It is so because of the process of photosynthesis. A closer look at a plant shows that it is firmly held in the soil by the help of roots. These roots absorb water from the soil and through the tiny tubes transport it to the stem and then to the leaves. The stem holds the leaves in such a way that they absorb maximum energy from sunlight. A leaf is a very important part of the plant. Photosynthesis, the ultimate provider of food and oxygen, occurs in the leaf. A leaf consists of a flat, thin lamina, which is joined to the stem by a petiole. The flattened lamina provides large surface area for trapping sunlight. A cross-section of the leaf shows the upper and lower layers of cells, which are flat and irregular. Below the upper layer is the layer of tightly packed cells. These cells have specialized organelles called chloroplasts. Chloroplasts contain the pigment chlorophyll, which gives the green color to the leaf. The next layer has loosely arranged cells with large air spaces. The section also shows tubes specific for transporting the absorbed water into the leaves. The lower layer has many pores called stomata. Each stoma is surrounded by two guard cells, which regulate opening and closing of the stomata. A leaf absorbs carbon dioxide from the environment through these stomata, which, after passing through air spaces, enter into the chlorophyll-containing cells. Both water and carbon dioxide molecules are finally absorbed by the chloroplast. These chloroplasts also have the capacity for trapping energy from sunlight, and this energy is used to synthesize glucose and oxygen from carbon dioxide and water. These oxygen and glucose molecules diffuse out of the chloroplast. The glucose is transported through other specialized tubes to all parts of the plant, aiding in their growth. The oxygen present in these air spaces diffuses out through the stomata. So, during photosynthesis, oxygen is given as the life-sustaining gaseous component of the atmosphere, while glucose or food is transported to all parts of the plant and used for growth. To summarize, green plants can make their own food by the process of photosynthesis. Plants are held in the soil with the help of roots. These roots absorb water and send it to the leaves through specialized tubes. The lower layer has many pores called stomata. A leaf absorbs carbon dioxide from the environment through these stomata. Both water and carbon dioxide molecules are finally absorbed by the chloroplast. Where in the presence of chlorophyll and sunlight, they synthesize glucose and release oxygen. The oxygen diffuses out through the stomata. The glucose is transported through other specialized tubes to all parts of the plant, aiding in their growth.
Okay, so that is not necessarily the elementary version of photosynthesis, little couple steps up, but it's not exactly like the PhD version of photosynthesis. There's a lot of reactions that happen. There's a lot of stuff that happens with photosynthesis, but that's the gist of it. Okay, and so understanding what these plant parts are, inner plant parts are, and the cells are within the leaf is an important piece. So in that video, we went through several of them, but to go through, we're going to be starting with the cuticle. Okay, so the cuticle is on the upper and lower sides of the leaf, okay? Of course, around the margin as well. Typically, it's waxy on top. Now, this is an observable trait. Okay, so this is something we can see on the outside of the plant. So when we're looking at plant leaves, in previous lectures, we've talked about identifying leaves by their shape, identifying leaves by their margin, identifying leaves by whether they have pubescence or not, right? There's all sorts of different ways to identify a leaf. There's also the ability to identify a leaf by some of the cuticle characteristics. And this note here, it says typically waxy on top, and it is a water impervious layer. Okay, so that typically waxy on top, if you think about it, you've probably held leaves or looked at leaves that have a waxy or shiny appearance. Sometimes it's not shiny. Sometimes it's more of a matte look to it, so it doesn't reflect a whole lot of light, but it is quite waxy. And we see this with leaves such as oak leaves. Many of the oak leaves have a waxy cuticle. A lot of the native plants have waxy cuticles, and a lot of times a waxier cuticle is generally associated with plants that have evolved in an area where there's lower amounts of water. Okay, so if you think about a lot of California native plants, we live in a Mediterranean environment or Mediterranean climate, and the Mediterranean climate is characterized by long wet winters and long hot dry summers. Okay, we get our water in the wintertime, and in the summertime we get little to no precipitation. Okay, of course, there's you know, the higher Sierras will get rain and the coast will get fog. Different areas of California could get some rain here or there, but in general, we have very dry summers, and the California native plants need to be able to survive those long hot summers, and so they need to manage the moisture within their structure to not release any water. Okay, so it's a water impervious layer, and that's the cuticle. Okay, now just below the cuticle is the epidermis. The epidermis, there's an epidermis on the upper and lower sides of the leaf. Okay, and it forms a boundary between the plant and the external environment. Okay, so it's a layer of cells that forms a boundary between the inner plant structures, where in this case, in the leaf, photosynthesis is occurring, and the outside, where there's heat or moisture or cold. So it's a protective layer. We have an epidermis, right? We have an epidermal layer, that's our skin. And so, and our skin does the same thing. It protects the inside of our body from the outside environment. Okay, so the epidermis protects against water loss. It regulates gas exchange, and we'll get to that, although that video went through it briefly. It can absorb water and mineral nutrients. Okay, so this happens especially in the roots. Of course, roots have an epidermal layer, but it's a little bit modified to be able to absorb water and mineral nutrients. Okay, now we will get to the stoma. There's a note here. Within the epidermal layer, okay, generally it's on the bottom of the leaf in the lower epidermis. Sometimes it's in the epidermis itself. Sometimes they occur in other areas of the plant, which is pretty interesting. But one thing that the narrator in that previous video mentioned were the stoma. Okay, so within that epidermal layer, there are cells called guard cells, and these create the openings that are called stoma or stomata, and sometimes they're called stomates. Okay, so the stoma are very important. We will get to that, but it's important to understand that they are specialized epidermal cells within the epidermis, and those are the guard cells. Okay, now the stoma or stomata are not cells, they are openings. Okay, the opening or the pore is the stoma. The guard cells on either side of the stoma are the plant parts. Okay, the stoma is just the opening. Now, below the epidermis, if we're going from the top down, okay, we have the cuticle, the epidermal layer, those are cells, and then we have a mesophyll layer. Okay, from here to here is the mesophyll layer. Let me see if I can get a better pointer here. Annotate, perfect format. Just want a mouse spotlight. That works. Yeah, that's better. Cool. So epidermal layer, upper epidermis, lower epidermis, and the mesophyll layer is sort of what makes up the bulk of the volume of a leaf. Okay, and it's broken into two different sections. The upper section is the palisade mesophyll, and the lower section is the spongy mesophyll. The palisade mesophyll, or the palisade cells, have tons of chloroplasts in these cells. And remember, within the chloroplast, that's where we have the chlorophyll. Okay, and that's what makes plants green. And it's actually interesting because they're not necessarily green, but they reflect green light. Okay, and that's why we see plants as green. Of course, not all plants are green. There's purple plants and yellow plants. Well, plants that are almost black. There's pink. There's a bunch of different cool colors of plants. That doesn't mean that those plants don't photosynthesize because they don't have chloroplast and chlorophyll. It just happens that the chloroplasts reflect a different colored light. And that's the only reason that they're a different color. They still function in the same fashion. This palisade mesophyll, or these palisade cells, are where the majority of the photosynthesis happens. Okay. Now the spongy mesophyll cells, the spongy cells, have air spaces between them. Okay, so this is where all the gases exchange that happens during photosynthesis. This is where that happens. And the majority of the photosynthesis, gaseous exchange happens in spongy mesophyll. And within the spongy mesophyll, there are mesophyll cells, and those do photosynthesize. Okay. There's also the palisade mesophyll. There's just more of them because they're so well organized. And that's where the majority of the photosynthesis actually takes place. There's the gaseous exchanges that are happening down in the spongy layer, but those gases come up and interact with those mesophyll, with those palisade cells as well. Okay, so this is the main process of photosynthesis. It happens in the mesophyll layer, and the mesophyll layer is essentially between the upper and lower epidermis, or vice versa, whichever way you want to look at it. Now, within the cells, within the leaf itself, there's also the veins. And remember when we talked about parallel and netted venation and identifying dicot plants by a midrib or sub-midribs or whatever type of venation they have, right? So those veins that go through the leaves that a lot of times we can see with our naked eye without any magnification, we can see those veins, okay? So those veins, okay, the veins themselves have both xylem and phloem that run through them, okay? So the phloem transports sugars or glucose that's produced through photosynthesis to the rest of the plant, okay? And that glucose that's created during photosynthesis goes into water and then flows through the rest of the plant. The elementary version of photosynthesis is that during photosynthesis, sugars are created and they're sent down to the root for storage, right? And that's kind of where the elementary version stops. The reality of it, a couple layers up from that, but not quite to PhD level, the reality of it is that phloem, right? Those phloem tubes carries glucose throughout the plant for all sorts of different plant processes. It also sends it down to the roots for storage and function. And it does a couple other really, really interesting things in the roots that we'll get to later in the semester. Okay, right now we just want a decent handle on photosynthesis and the inner plant structure. Later, we'll get into the weeds on, not literally, we'll get into the weeds on carbon storage, carbon sequestration, and plant exudates, which is like a fascinating process. Anyways, phloem is two-way. The phloem will go through the phloem tubes, okay, and that is essentially sugar water, right? that flows throughout the plant for whatever other types of plant functions need the glucose, okay? That's the plant's food. When pests like aphids, okay, piercing, sucking pests like aphids, scale, etc., when they are on plants, what they're actually trying to eat is they're trying to tap into that phloem. They want the glucose. Okay, the glucose is what supports them. And if you think about aphids, we've probably all experienced aphids, at least seen them. They are on those plants during heavy vegetative growth, and they're piercing into that plant and they're trying to pull out the glucose. They're trying to pull out that plant's manufactured, self-manufactured food. And the result of aphids is honeydew, right? We have a lot of honeydew that falls onto the ground below plants that are aphid infested or have some sort of infestation of aphids. And that's essentially what that is. Okay. Now, the xylem is a one-way tube. Okay. This is where water is transported with nutrients from the roots to the leaves for photosynthesis and respiration processes. Okay, so the xylem is sending water with water-soluble nutrients into the leaf for this photosynthetic process. Other things too, but that's primarily it. Now we're going to talk about that stoma. Now the stoma, I've always been sort of fascinated by the stoma. It's a really cool part of the leaf. It's interesting. It's really cool looking. But it really controls a lot of the plant's respiration. Right? So stoma are minute pores in the epidermis of the leaf or stem of a plant. Okay, remember I mentioned that sometimes those stoma might not just be on the leaf. Okay, generally we think of stoma as being on the lower epidermis. Okay, we think of it on the lower epidermis. But there's some plants that have stoma on the upper epidermis, lower epidermis. There's some plants that have stoma on the trunk, right? On the stem of the plant. Okay, so this can happen really anywhere. When they are on the trunk or stem of that plant, if you think about it, if the stoma are on the stem of the plant, that means that that gaseous exchange happens on the stem of that plant, right? And so that means that there's some mesophyll within the stem of that plant. Quick example. Cercidium. It's a really popular plant right now. Get into images. This is not my normal. So this is, I guess let's look at the plant itself because we like plants, right? So let's look at let's look at a plant. There we go. This is the Cercidium, southwestern native. The really popular one right now is called Desert Museum. It's a triple hybrid. There's 3 different Cercidiums that were hybridized together to create the one that's called Desert Museum. You go into any nursery these days, and it is kind of the most popular Cercidium. So the Cercidium is a really, really cool plant. Clearly, it's a very low water use plant. Has beautiful yellow flowers in the spring. Very, very fine textured plant with a beautiful green trunk and stems. Now, one of the really cool parts of this plant, well, beyond like the overall appearance, which is super cool, but you can see this green stem. Now, I mentioned in a previous lecture, Aesculus californica, the California buckeye. Remember, it's drought dormant. It's drought dormant. And so as soon as there's not enough moisture in the soil and the air during the summer, California buckeyes drop their leaves and they essentially just go dormant for the rest of the summer through the winter, and then they come back in spring. So they have a very short leaf out period. Now, Circidium does something similar, but it does it in a little bit different way. So it is drought deciduous. Okay, so as soon as there's like minimal amounts of water, and it has to get pretty low for Circidium, but as soon as there's minimal amounts of water, it will drop its leaves, but its stems can photosynthesize. Okay, so it essentially drops all of the material off that has a great potential to lose water, and it just goes into a slow respiration while photosynthesizing only in its stem. Okay, cool, nerdy stuff, but cool stuff. Okay, so the stoma, gaseous exchange, stomatal opening and closure helps in the gaseous exchange between the plant and surrounding environment. It helps to in transpiration and removal of excess water in the form of water vapor. Stomatal closure at night prevents water from escaping through pores. Okay, now this is a generalization, right? We're looking at generalization. Most plants do it this way. We're going to look at some examples of some alternate times when stoma are open, okay, which is also kind of interesting. So it maintains the moisture balance according to weather by opening and closing. So essentially, those guard cells, okay, those guard cells that are responsible for being turgid, right, or open, okay, when there's plenty of water, those cells are filled with water and they're turgid, so they're rigid. And when they're rigid, it opens the stoma. The stoma is open. Now, when there's not enough water in the plant, the guard cells are flaccid. Okay, so they're essentially empty and they close the stoma. Okay, so it's very water dependent, and the plant will, if it gets too hot and there's not enough moisture, the plant will close up its stoma and essentially deflate those guard cells so that it's not losing water through those stomatal openings. And when water vapor is lost through the stoma, that's called transpiration. Stoma facilitate carbon dioxide uptake and release of oxygen during the process of photosynthesis. In that process, right, the main goal of that stoma, the main reason that stoma exists is when it's open, carbon dioxide can come into that mesophyll layer and do the gaseous exchange, and the oxygen goes out. But in that process, water vapor is also lost. Okay, so that is transpiration. Water vapor is lost through those openings as well. Okay, so it's not incredibly efficient because there's a couple flaws in the system. So this is a quick video specifically just on stomatal openings.
Process of stomatal transpiration. Most of the transpiration takes place through stomata. Stomata are microscopic parts consisting of two kidney-shaped guard cells. In stomatal transpiration, water vapor moves through the stomata of leaf. Water absorbed by the roots rises through the stem and reaches the tissues of the leaves through veins. A large number of spongy mesophyll cells in the leaf have their surfaces exposed to the intercellular spaces. The surfaces of the cells give out some of the water, which forms a thin film. The water from this film evaporates, and the water vapor formed saturates in the intercellular spaces. This vapor then diffuses into the other connecting intercellular spaces and finally reaches substomatal space, from where it escapes out through the stomata. Stomata are absent in the submerged hydrophytes, that is, Hydrilla, Vallisneria. Mechanism of opening and closing of stomata. Transpiration occurs as long as the stomata are open. It stops when they are closed. Open and closed stomata. Closed stomata. Flaccid guard cell. Stoma closed. Stoma open. Turgid guard cell. Thick cell wall. Thin cell wall. The opening and closing of stomata depend upon the turgid or flaccid state of guard cells. When the guard cells are in the flaccid state, stomatal aperture closes. During daytime, as a result of photosynthesis, as the guard cells have chloroplast, concentration of carbohydrates rises, leading to osmotic uptake of water by the guard cells to swell. Since the wall of guard cells towards stomatal opening is thick, while outer convex wall is thin, inflow of water in the guard cells causes them to bulge outwards, thus widening the stomatal opening. When the osmotic pressure of the guard cells becomes lower during night, the water leaves these cells due to exosmosis and moves to the neighboring epidermal cells, having cell sap of higher concentration. The guard cells become flaccid and shrink, and the stomatal aperture closes.
So these guys keep talking about this only happens during the day, right? And then the night they close up. But this fella here is going to tell us a little different story.
Okay guys, welcome to our screencast. We're going to be talking about some different types of photosynthesis. I know you thought you'd learned all there was to do about photosynthesis, but there are a couple of different types we're going to look at. C3, C4 and CAM photosynthesis. And although these are really complex reactions, we are just going to have a look at the summary of the differences of some of these reactions. Now these different reactions are adaptations for photosynthesis, and as you know from studying adaptations, they can be structural, physiological or behavioural, and they can occur in both plants and animals. And the adaptation we're looking at here is a physiological adaptation in plants. That's how the reaction actually happens in the cells. Now photosynthesis is all about the sun and capturing that light energy from the sun. And when this happens, we have a problem because okay guys, welcome to our screencast. We're going to be talking about some different.
Sorry about that.
We are just going to have a look at the structural, how the reaction actually happens in the cells. Now photosynthesis is all about the sun and capturing that light energy from the sun. And when this happens, we have a problem because carbon dioxide is needed for photosynthesis, and that gets in the stomata through the leaves, in through the tiny pores. And when those stomata are open for the carbon dioxide to go in, we often have a loss of water, and that's through transpiration. When the stomata are open to let the carbon dioxide in, we have the problem that the plant can lose water. And the adaptations in photosynthesis are for the plants to get around this process by adapting the chemical reaction that occurs. First, let's look at C3 plants, and these are most types of plants, and they're called C3 plants because the first compound they form in this stage of photosynthesis is a 3-carbon molecule. And the problem that happens is that quite a lot of the carbon dioxide that's taken in by the plants is actually lost because this enzyme also reacts with oxygen. And this is fine in mild conditions where the stomata can stay open day in, day out, and the carbon dioxide can easily get into the plant, and water loss probably isn't a big issue. So let's look at C4 plants, and two great examples of C4 plants are maize and sugarcane. So they're called C4 plants because the first molecule is a carbon molecule, and the benefit here is it doesn't react with oxygen like in C3 plants. So this means that the trapping of the carbon dioxide is a lot more efficient, and the stomata don't need to be open. In C3 plants, the stomata needed to be open for such a long time because the carbon dioxide, the trapping of the carbon dioxide wasn't too efficient. Whereas in these plants, they can just open them for short periods of time because they've got a really efficient way of trapping that carbon dioxide by creating this 4-carbon molecule first. I suppose the only real downside is it uses a little bit more energy, but certainly when it's hot, it's an adaptation for these plants, and they're a lot more efficient in overall photosynthesis and their survival by using this method. So to just look at C4 plants in a little bit more detail, is that 4-carbon molecule is created from carbon dioxide, and that happens in the mesophyll cells of the plant. And that molecule is then transported to the sheath cells of the plant, and that's where it's converted to a 3-carbon molecule, just like C3 photosynthesis, and the process continues very similar to what you'd see in a C3 plant. An alternative adaptation that plants have to hot conditions is using CAM photosynthesis, and some classic examples here are a whole range of cacti and desert plants and pineapples, and that again it works well in hot conditions but particularly in dry conditions, and this is well suited for plants that only open their stomata at night time. During the daytime it's just too hot for them to have their stomata open and to be losing all that water through transpiration through the stomata. So the way that they undergo photosynthesis, at night time they have the stomata open, they take up as much carbon dioxide as they can, and they convert it into a 4-carbon molecule which is stored. In daytime the stomata close, and this 4-carbon molecule is then used in normal C3 photosynthesis, and the plant pretty well goes through the same process.
Okay, so there we have it. It is not just the general open stoma during the day and closed at night. There's all sorts of different really interesting adaptations that plants have made that are influenced by the environment that they're actually growing, right? So cactus and succulents, for instance, right? We think of cacti and succulents as very low water use plants. Okay, not all, but the majority of them are. And they have CAM respiration, okay, CAM photosynthesis, where they essentially they open up their stoma at night. They store that energy from the sun, and then they open up their stoma at night, and that's when they're photosynthesizing. Okay, so really, really interesting stuff there. Again, high level, broad strokes on it, not getting super deep into it, but important to know that there are some differences there. Okay, so that takes us through all of our info about general leaf structure. Now we're going to get into stem structure. Okay, this is basic stem structure. Looking at the stem itself, the function of the stem is support and transport. Okay, support and transport. And what we're going to be looking at through the majority of these is this diagram. Okay, now this diagram shows primary growth and secondary growth. The primary growth is essentially the first year of a seedling. Okay, and these, I guess let's clarify. We're looking at dicot plants right now. Okay, remember there's some differences in dicot and monocot stem structures and their vascular bundles. Remember the vascular bundles, vascular bundles, vascular bundles, vascular bundles, vascular bundle. These are all organized in essentially concentric rings, and they're well organized, whereas monocots have a little bit more random layers through them. Okay. And this is what I'm talking about. So that monocot versus dicot. Here we have the dicot. Okay, we have vascular bundles that are arranged in essentially a concentric ring. Okay, whereas the monocot has vascular bundles primarily on the outside, but it has them randomly scattered through what would it be essentially like the equivalent of the pith of a dicot. Okay, so we'll get into this question in a couple minutes. Can you graft a monocot? Generally, the answer is no, but there's different things that can be done. So back to this diagram and looking at the different parts within the stem in this cross section. First, we're looking at xylem. Okay, so we have some primary xylem and some secondary xylem. Okay, and the xylem does the same thing as within the leaf. It's for transport of water and nutrients from roots to the leaves for photosynthesis and respiration. Phloem, just like within the leaf, transports glucose produced through photosynthesis to the rest of the plant and to roots for storage and function, and the rest of the plant for different plant functions. Okay, so we have, again, that primary xylem and secondary xylem. And then we have primary phloem and secondary phloem. Now, we have some cambium layers within the stem. Okay. Now, the cambium layers are another interesting part of the plant. Just like, well, not just like, but similar to the apical meristem, right? Remember, we talked about the apical meristem and those meristems, right? Where meristematic tissue is, it's stem tissue, it's undifferentiated cells, okay? And these cambium layers are these very, very microscopic, very small, thin layers of undifferentiated cells that are actively dividing, creating xylem to one side and phloem to the other. So we actually have two of those cambium layers. So one of those cambium layers essentially is immediately between the secondary xylem and the secondary phloem. And what that layer is doing is, as it's growing, as that plant is growing, it's creating xylem cells to the inside of the plant and phloem cells to the outside of the plant. Now, there's another cambium layer within a plant, and that's the cork cambium. Okay, that's the cork cambium. So the cork cambium, it's also a microscopic layer of undifferentiated cells actively dividing and creating, in this case, phloem cells to the inside of the plant and epidermal cells, or if we consider a tree, bark cells to the outside of the plant. It's creating that epidermis. Okay, so again, two very interesting little spots on the in this cross section of a stem. Cambium layers. The pith. The pith is the inside, the inner side of the stem cross section. And this is, it depends on the plant itself. Sometimes the pith is very hard, sometimes very open. But within the pith, they're all spongy cells, so generally a decent amount of pore space between them. And these are for storage of carbohydrates. Okay, so that really takes us through the bulk of what the inner parts of a stem are. Got this little ditty for you. This is a bud graft. Oh, started way too late. Sorry about that. I'll speed it up here. Okay, so they're cutting into the epidermal layer. And they're separating it right at the cambium. Separating the epidermal layer from the cambium. And what's right on the inside of that is the phloem. Now what they're doing is they're taking a bud from a different plant cutting it to size. Remember, there's meristematic tissue at the bud. And they're sliding it into that tissue, making contact with the phloem. It's a really basic bud. Okay, so the bud grafting, that's what they just did. So essentially, what they're doing is they are separating that epidermal layer at the cambium, and they're sliding a different bud within that. And the goal is to get them to grow together. And what these people did was they basically got a different rows right up. Okay, so it's pretty fascinating stuff. So along with that, let's look at some other cool things about that epidermal layer. Quercus suber, or the cork oak. Okay, there's a lot of them that grow in the Sacramento region. Grows really well here. It's a true Mediterranean basin native, but it is an oak. It is a quercus, and it survives incredibly well here once established with no supplemental water. Okay, now we know that corks are made from the cork oak. Of course, nowadays corks are also made from plastic, but generally, I guess there's a lot of screw tops now too. But anyways, corks are made from cork oak. Okay, now the process is that they strip the epidermal layer, okay, that cork layer, off of the tree, leaving the cambium layer intact. Okay, they leave the cambium layer intact. They only remove the cork portion or the epidermal portion of that plant. And throughout the years, the cork has been developed at the cork cambium, where to the inside, it's creating phloem, and to the outside of that cork, of that cambium layer, it's creating this epidermal layer, which is the cork. Okay, so and we know this because you, on a mature cork oak, you can essentially harvest the cork about every 7 to 9 years. Okay, about every 7 to 9 years. So fascinating stuff, a lot of interesting plant parts. And that takes us through the basics of our leaf structure and the functions, and our stem structure and the functions. Okay. Thank you.